Hi, I'm Brian Foley from Los Alamos National Laboratory, HIV sequence and immunology databases. And I'll be telling you about our sequence database today. Tomorrow, my colleague will tell you about the immunology database. Share my screen. Point, share, slideshow. So our databases were founded in the late 1980s by Gerald Meyer, who recognized the diversity of HIV, and he knew right away that we would need a database for this virus because it was so diverse. Today we have 7 other employees besides myself, and tomorrow Elizabeth Sharon Fong will be telling about the immunology side of the database. We have millions of HIV sequences, actually only about 1 million sequences right now, and hundreds of epitopes, both antibody epitopes and antibodies that are known to bind to HIV, and also Ctl and helper T cell epitopes. Today, I'll be giving you an introduction to the HIV sequence database and a little bit of introduction about HIV in comparison to other viruses. And then I'll explore HIV evolution, sharing one GAG epitope as an example. So you get to our database at hiv.lanl.gov, or you can use www.hiv.lanl.gov. Brings you to our entry page. We have a sequence database, an immunology database, and other viruses include Hep C, hepatitis C virus, human filoviruses or hemorrhagic fever viruses, and SARS-CoV-2 coronavirus database now. It's actually not a database, but a database analysis tool. So once you're in the HIV sequence database, you can go to the search page through a pull-down menu. You can enter the search interface through these links. We have several different search interfaces. One where you search by geography, one that I'll show you in a second where you search on many fields. And then there's a Genome Browser that I'll describe also. Under our guides, we have tutorials, explanations of circulating recombinant forms of this virus, a HIV gene map, and so on. And if you click on tutorials, you can come to this list of his database presentations from other conferences where you can get a lot more information about how to use our tools. Today, I'll be covering just a couple of our tools. And you can always email us if you have any questions. We do respond to that email address. On the main search interface, there are links to tips and tricks of how to get better information out of searches. You can see from the main search that these are the most common fields that people search for. And down below, you can open up additional windows that will be similar to these with many different types of information. You can search on publication information, you can search on the PubMed ID number, the year of publication, author name, and so on. Or you can search, up here you can search on country, but down here you can get more geographical information. You can search by region of the world, like Africa or Asia, and so on. Under the tools menu, we have many tools. Some are specific for HIV and CIV, CIV being simian immunodeficiency viruses, which are all the primate immunodeficiency viruses. There are tools that are have some HIV or CIV specific features, but can be used with other organisms. And then anything colored in blue can be used with any organism. Any alignment of DNA or protein sequence or DNA or protein information is the same for any organism at some point. All viruses evolve, but each virus has its own unique epidemiology and evolution. The epidemiology often influences the evolution. HIV, hepatitis C virus, and some other viruses persist in one host individual for a long time after their host's immune responses mature. Then the virus can be passed down to a recipient of the virus long after the initial host has driven some evolution of that virus. That virus may have escaped a strong antibody response or escaped a Ctl response in an individual, and then the escape mutant could be passed on to the next recipient. Influenza viruses, coronaviruses, common cold viruses, and so on typically infect one naive host, and rapidly passed on to the next recipient before any immune selection pressure has taken place. So HIV has been infecting humans for 100 years or more and is relatively slowly spread around the world. Hepatitis C virus, likewise, has been infecting humans for more than 20,000 years, and likewise spreads around the world quite slowly. Whereas the influenza and coronaviruses can spread around the world in a matter of weeks, as we've recently seen. And one particular strain of the virus, such as Delta variant of the new SARS coronavirus, can replace other virus or other strains of the same virus within a matter of weeks or months. So I went to the IEDB and searched on Ctl epitopes for HIV-1 just to see what would pop up. And on the very first page, I noticed many variants of the Slint-Vattel epitope. And I had heard many stories about Slint-Vattel over the years of me studying HIV, but I'm not really an immunologist. I'm more of an epidemiologist, but I've heard a lot about Slint-Vattel. So I thought, great, it's a common epitope. It appears in the IEDB, and I'll explore how variable this epitope is in HIV-1 and how it varies over time. So one of the first things I was interested in is going to the Genome Browser to find where this epitope is in the genome. The Genome Browser we created so you can scan across the HIV genome or individual genes and zoom in on regions and see what we know about them. So I went to the Genome Browser. I knew the epitope was in the GAG protein, so I select the GAG protein view. And within GAG, I entered Slint-Vattel up here in the search window to zoom in on that region of the protein. And now I can see from the epitope, the GAG map up here, that I'm in P17 around amino acids 77 to 81 or so. And you can drag in these other tracks to the view. And what I've dragged in is the entropy of subtype, or entropy within subtype B clade, entropy within subtype C clade, and Ctl epitopes and HXB2 sites of interest. HXB2 is the reference genome we use for HIV-1. Anyway, you can see that there have been numerous epitopes of Slint-Vattel, numerous Ctl reactivities to Slint-Vattel have been noted, mostly with human AO2 or A2 genotype, human genotype. You can also see that this SLY, the Y, is quite variable, has high entropy in both the B clade and the C clade. And some other sites nearby have variable entropy, so they're less variable in the B clade than they are in the C clade, which is not unusual. Most regions of the genome have more diversity in one clade than another. It's not unexpected. So now going to the immunology database, I searched on the epitope Slint-Vattel and find out more information there. So I found out we have 301 matching records that include Slint-Vattel. Each one has a link to an epitope alignment. You can get to a GAG epitope map and so on. In this entry here, the epitope is defined as being a little bit broader, which is not unusual. A lot of people used 15 mers for the reactive peptides, and they know that the epitope is somewhere within that 15 mer, but don't know exactly what it is. Other ones have narrowed it down to a 9 mer. You can see the Slint-Vattel right here in the middle. And this is not only showing you what's diverse, like the SLY, the third position there, we saw that that was diverse, but here we're seeing it's diverse only in two amino acids, phenylalanine and tyrosine. So using QuickAlign to look at the Slint-Vattel epitope or analyze a line, it looks at an alignment of that region of the GAG protein in all of the entries in our database that were one sequence per patient, and they had to be complete GAG gene and so on. You can see we had 6,144 sequences in the alignment we used. And again, you see that the F versus Y at the third position is common. And over here on the other anchor side, we see threonine versus valine. You can get the exact breakdown. Phenylalanine in all subtypes was 48.7%, and tyrosine was 46.8% here. And you can also get a breakdown by subtype. So here we're looking at subtype B, and we can see that in subtype B, the tyrosine outnumbers phenylalanine by about 60% to 40%, 37.5 there. So you can get a breakdown of the number of counts per subtype, and you see we had almost 2,000 subtype B sequences that we're analyzing there. So looking at the alignment from which those statistics were gathered, you can see a protein view. I'm using AliView here on a Mac to display the protein view or the DNA view of the same region. So we see that the Y versus F at the SLY Slint Battle site is in this region of the subtype B alignment, which is mostly from China. The phenylalanine is outnumbering the tyrosine here. And you can also see that it's a TTC to TAT change, which is a transversion in the DNA sequence or RNA sequence of an RNA virus. And transversions are generally quite rare in comparison to transitions. So apparently a T to C mutation here to make a TCT codon is lethal to the virus, or at least selected against. So we're seeing only the phenylalanine and tyrosine being allowed at this position. Likewise, over here, we're seeing a dual mutation of AC to GT quite frequently. And that's a little bit unusual. Again, it's indicative of selection pressure when, you know, the GCC codon is not seen, but we see a GTC quite often. So another part of the quick align or analyze align analysis shows you how many amino acids have changed in the average virus in this epitope. So we see that a huge percentage, 24% of the sequences are identical to what we typed in Slint Battle. 37%, almost 38%, have one mutation only in this epitope. It might be the F or the V. And then 25% have two mutations, and very few have 4 or 5 mutations in this epitope. And years ago, we noted that many of these sites seem to be toggling back and forth, like F to Y, Y back to F, and V to T, V back to T. We named those toggle sites and noted that this happened at numerous places, not just in Ctl epitopes, but throughout the genome. This is very common for only a couple of amino acids to be present. So that was the birth of our Mosaic vaccine design tool, which designs 2 or 3 or 4 synthetic viruses that have natural sequences like Slint Battle or the whole natural GAG protein encoded, but they have a combination of these mutations such that you cover all of the diversity seen in the viruses with very few different vaccine strains put into the vaccine. So a PubMed search for Slint Battle hits only 55 papers, whereas our database has 301 records. And that's partly because people will sometimes refer to this as SL9 for S beginning, L at the end, and 9 amino acids. So Slint Battle or SL9 are common names of this epitope. Also, the epitope may not be described in the abstract or easily searched PubMed search. They may be more in the discussion of the paper. Okay, so after looking at the alignment where we had 1 per patient, I went to an intra-patient search and searched for sets where patients had been followed over time. And I wanted to see if this epitope was evolving within each patient over time, highly variable within each patient, or did it only slowly evolve and variable between patients? So here I've only put in light lines, but you can see there's 1, 2, 3, 4, 5, 6 patients represented here, and only one of them has some variability in this epitope. And it's not in one of the anchor residues at the side, not the Y or the T or V over here, but in the middle of the epitopes. That could be an escape mutation, but more likely it's just some natural variability, and it's a leucine versus a valine versus isoleucine change, which is quite conservative. But here you see between patients, we have some of the Y form and some of the F form. So anyway, apparently it's not evolving quickly in all patients. These patients have been followed for a few years each, and most of them showed no change in this epitope. So then I went to a cohort, a Korean hemophiliac cohort, where I knew that a single strain of the virus from a single donor had been transmitted into 20 different hemophiliac patients. And those patients have been followed over 20 years now. So this is a huge amount of time, several different patients, 20 different patients. And I was looking for variability again. And again, we see not really a lot of variability happening. The F versus Y third position did happen in a few patients, but you can see right here that the infecting strain, the donor, already had phenylalanine there. So it's possible that this is already an escape mutant or something else is going on. But again, I was quite surprised not to see much diversity in this quite long followed cohort of patients. Subtype A6 of HIV-1 has been spreading through the former Soviet Union since the mid-1990s. So here we're looking at, over 30 years of evolution. And the infecting strain in this case had a tyrosine in the third position. And again, we see a little bit of conversion to phenylalanine, but not a lot among all these patients. There's a couple of possible guesses as to why that might be. One is that this is primarily an intravenous drug user epidemic. So maybe the virus is being passed from one person to another more rapidly than you see in a heterosexual or homosexual epidemic. Typically, IV drug user epidemics spread a little quicker, and the virus tends to be passed more like a influenza virus, where it's passed before much selection pressure has taken place. So again, not as much diversity as I was expecting to see. So then I went to the immunology database again, and I looked for escape variants to find out maybe if I could pin down some of the patients where this is known to be an escape variant, and have a look at those patients followed over time. So I picked this Carlson 2007 paper, and it has a link to the sequence database entries in there, and I got those out and aligned them and started having a look at that. So the paper had a phylogenetic tree made from their sequences, and a major point they made was at some point the tyrosine mutated to phenylalanine here, and most of the viruses in this subclade within this one patient, OP428, tended to have the phenylalanine. So the story might be there that this patient was infected with the tyrosine containing strain. At some point, this epitope was under selection pressure, mutated to phenylalanine. So later on in the patient's infection, had a dominance of phenylalanines in their phenylalanine containing strains in their blood. And that all looks pretty reasonable, but I built my own phylogenetic tree to have a look at this. My tree has basically the same features as the tree that was published, which is confirmatory and expected and nothing interesting really. But as I was looking at my tree and the tree in the published paper, I noted that the tyrosine to phenylalanine change wasn't really consistent. It's not like all of these guys have phenylalanine and all of these have tyrosine. There's some flipping back and forth, or that toggling that I talked about before. Now, if we trusted that this was a true phylogeny and this is the way the virus truly evolved with no recombination, then we would say that that single site had to be mutating back and forth between tyrosine and phenylalanine. But the other explanation potentially is that there's recombination going on in this patient. So that's quite likely, is that some of the viruses have recombined. However, some of these sequences were sampled fairly early. No, that's actually week 63. That's not very early in the infection. These are week 79 mostly here, and some week 63. Anyway, so the ones fairly late are the ones that have phenylalanine, whether they're within this late clade or not. Again, that's suggestive of recombination happening within the patient. Zooming in to look at the alignment there, we see I've got some of both patients here. OP428 is the one with the OP428 is the one with the selection pressure happening, and OP599 did not have so much diversity in the epitope. But OP599 already had phenylalanine and a few other mutations. Perhaps he was already infected with the escape variant that wasn't reactive at this epitope. And again, 428, you see again, it's the TTT to TAT transversion mutation happening. Another tool we have is the protein feature accent tool. Now you can look at the 3D structure of the GAG protein, and you can add your alignment down below, highlight an region of interest. Here I've highlighted the Ctl epitope, and I can see that it's in this coiled coil in the center or core of the GAG P17 protein. And that's, it's very usual or typical for the core of a protein, and especially an alpha helix that has to be preserved to maintain the structure of the protein, can be quite conserved. So that, it doesn't surprise me that this epitope would be conserved. It's in the core of the protein. You can, when you're viewing this on the web, you can actually rotate the molecule, flip it around. You can change the cartoon to ball and stick or space filling and so on. You can pick other color schemes and so on. Anyway, it's a kind of a quick and dirty. You probably wouldn't publish this image, but you can explore your protein structure and then publish an image that's made with a more sophisticated 3D protein drawing tool. But the advantage is you run this on the web. You don't need to download any special software. You don't even need to download the PDB file. We already have them loaded for you and so on. So one more level of diversity here is to look at this region of the GAG protein in all primate lentiviruses. So this will tell you what's happening over millions of years of evolution, or at least many more than 100,000 years of evolution between the African green monkey virus, the Debs colobus monkeys, drill monkeys, and so on, chimpanzees. And you can see over this huge amount of time scale, the Ctl epitope is fairly reasonably conserved. The S and a few of the other amino acids at least are quite conserved here. You can also see that they're maybe not quite as conserved as some other regions like this highly positively charged end of the protein. And that's all I have for you today, except I will show you one more slide where you can download today's lecture and some tools such as the AliView tool. That's a tiny URL, HipDB IEDB 2021. I'll put up the AliView and BioEdit and FigTree phylogenetic tree building software, and my slides and some sample data files such as those alignments I was showing you today. So thank you for attending, and if you have any questions, we'll have a question and answer session right after this. And if you have questions later, you can write to SeqInfo or Immuno. Thank you very much.